Okay. Hello, everybody. It's Friday, and we have another VTLS seminar. Today's speaker is Chloé. Lahondère. You're gonna need to apologize me because my French is really bad. And it's actually a pleasure to have her here. We tried to have her last semester, and unfortunately, her seminar was scheduled right after everything was canceled, and we needed to move her to this fall semester, and she moved around a couple of times until finally we managed to have her scheduled for today. So thank you very much for your patience. Chloé actually did her bachelor, master, and PhD at the University of François Rabelais in France. And her bachelor and master were in biology, PhD in neuroethology. After that, she moved for a postdoctoral position to the University of Washington in Seattle, and did a couple of years postdoc in neuroscience. Her expertise is in vector biology, and this has tremendous implications for diseases that they are related to or they are transmitted through these vector-bornes, and they don't have, for which they don't exist really vaccines. So her work is particularly important in that area. She has joined the Department of Biochemistry in 2017 and has been there since then. And her research focuses in studying thermal biology, ecophysiology, and neuroethology of disease vector insects and ticks. She does a lot of collaborative work, multidisciplinary and integrative, using a multidisciplinary and integrative approach. She combines field work and molecular biology, which I think is kind of a really cool part of her research, able to do something that goes just right beyond the molecular aspect and the bench work. She is a member of several professional organizations and is affiliated to the CZAP, which is the Center for Emerging Zoonotic and Arthropod-borne Pathogens. She's also affiliated to the Department of Entomology in Virginia Tech. She's a reviewer for several journals in her field, remarkable journals in her field, and she's currently funded by numerous seed fundings for her research from different organizations around campus. Without further presentation, and I apologize in my horrible French, I'm gonna leave the floor to Chloé.
Well, thank you so much, Carla, for this nice introduction, and your French is totally fine. I really appreciate the invitation to give this talk, and I know it would have been better to be with all of you, but at least we are safe, so that's the most important thing. Okay, so I'm going to talk to you today about mosquito-plant interactions, and that has been like a huge part of my research focus in the lab. So the thing is that we are also interested in many other aspects of disease vector biology and ecology. In particular, we are interested in relationships between mosquito and climate change, as well as developing new control strategies. So more like an applied kind of aspect of the work. What can we learn in the lab that we can then use in the field? And we are not only working on mosquitoes, but we are also working on other disease vector insects, such as kissing bugs and ticks, not ticks, tsetse flies, as well as other arthropods, such as ticks. And we are interested in interactions that they have with their host, whether they are warm-blooded animals, such as humans or birds, or cold-blooded animals, including frogs and snakes. So why is it important to study these insects? Well, mosquitoes that I'm going to focus on today are the deadliest animal on Earth. They kill more than 1 million people per year due to the different pathogens that they transmit to humans, including Zika, dengue, malaria, and yellow fever. And there are many challenges that are associated with these disease vector insects, and in particular mosquitoes. Like Carla mentioned, like vaccines and treatments are not available for several of the diseases that they can cause to humans. There is also a rise in the insecticide resistance that is challenging control strategies. And we have also mosquitoes that are kind of just outsmarting our control tools, including bed nets, for example. Like some mosquitoes have started shifting their biting behavior to the day, so they don't get caught in your bed net, for example. And obviously, like one of the big challenges is climate change and several invasive species that are kind of spreading all over the place. So what do we need? We need new solutions and new tools to limit mosquito populations and to prevent pathogen transmission. So how pathogen transmission works? Well, females are the ones that bite, and when they bite, they can transmit pathogens to their host, whether they are animals or humans. And they use the proteins that are contained in this blood to produce eggs. And that's pretty much it. But the thing is that, why is it important to, on the opposite, study sugar feeding? Well, sugar feeding, for example, is implicated or is for both males and females. And it's implicated in so many aspects of their biology. We've shown that it's increasing their flight and their activity. It's implicated in their metabolism. It's extending their longevity. And it's also increasing egg production. So if you are able to dissect the mechanisms that are underlying sugar feeding and use that for control tools or disrupt this behavior, then you have just like something that could be like really interesting to implement in the field. So where do they get these sugars? They can feed on ripe fruits. They can feed on honeydew that is produced by aphids. They can feed on tree sap, which is kind of super viscous, which has like some other kind of cool challenges that we are interested in learning more about, but that's for a different story. And obviously plant nectar is one of the main sources that they use. So today I divided my talk in 4 different parts, and all of them have like kind of this common thing where we want to learn what we learn from the lab experiments and what we learn from the field can just like inform each other, and the aim basically is just to have a better understanding of mosquito ecology and behavior, and ultimately develop this kind of new control tools to reduce mosquito populations. So let's dive in with the first part, that is about the interactions between mosquito and orchids. And all of this started was with this paper from Dexter in 1913 in Science. And so the paper is written like kind of a very interesting way. It's like just kind of a story where he mentions that his assistant was in the field, and so these mosquitoes kind of flying around with like this like weird like yellow masses on their head, and she was wondering what it was, and he went to the field and found out that it was actually pollen from an orchid. And so that was kind of like the first insight into mosquitoes not only being annoying and biting monsters, but also having like a part in the ecosystem, and in particular for like plant ecology. But since then, so since like more than 100 years ago, we don't almost know anything about the relationship between these orchids and these mosquitoes. And there is also an important part to get about this orchid, is that it's an endangered species that so far we know that only the mosquitoes are probably able to pollinate. So there is like a huge impact into getting to understand this kind of relationship. So in the mid-70s, there was a little bit of interest in this system where what we learn is how the pollination actually is working. So the mosquito is gonna like land on the lip and push its head inside the flower to get the nectar that is found in the spur here. And by doing that, these 2 structures that are there that are called pollinia are gonna be moving, and they are moving so much that they can basically fall, and at the base of each pollinium you have this sticky pad, and the sticky pad can get stuck on the mosquito eye, and then these mosquitoes are going to transfer the pollen to the next flower, and pollination occurs. That's all we know. We don't know anything about the signals that the mosquitoes are using to find these flowers, or what they even get from these flowers in terms of like nutrition. So what we did first was field work. So we did some field work in the Wenatchee National Forest. So this plant is found mainly in the northern part of North America. And there, the important thing was that we had access in a kind of a small piece of land to a large diversity of orchid species, because different ecosystems were present, including wetlands and forests and meadows. And definitely a lot of mosquitoes, which was great. This plant is particularly difficult to find. It's a very small orchid, it's green, it doesn't smell a lot, and it blooms only 3 weeks per year. So it was kind of a challenging project, but also like kind of really exciting. So we studied 6 different orchid species that are pictured here. They are all pollinated by different insects. They are, in some cases, also able to self-pollination. They have like different traits, including flower color that is very variable between the different species. And the scent, at least to the human nose, and human nose is actually me, is highly variable. Some of them have a very strong smell that you can smell like them from like several meters away. And some of them don't smell anything. Like the Ciliaris has this very tiny, small smell. They attract the butterflies mainly due to their like bright orange color. So the first experiment that we conducted was a field observation of interactions between the mosquitoes and these plants. We wanted to learn more about what kind of mosquitoes are visiting these plants, and are both males and females actually attracted by these plants. So indeed, both sex are interested in that plant. So you see a male here, like fluffy antennae, going in the flower, and then we found it with like these 2 pollinia attached and this kind of extended clear belly full of nectar. And you can see here this female also bearing these 2 pollinia on its head. And we found quite a large diversity of mosquito species visiting this plant. It was largely dominated by these Ochlorotatus communis that are like Aedes mosquitoes, along with Ochlorotatus increpitus. And both males and females were attracted to that plant, which was not known before. The next step was to try to understand the exact role of the pollination by these mosquitoes on the fitness of the plants. So we did some pollination exclusion experiments where the plants are bagged in the field before they are blooming. And the reason why we do that in the field is because, again, this plant is endangered, so there is no way you can take it back to the lab or that you can find it in nurseries. So almost all of this study was conducted in the field. So we buy the plants and then we count the number of fruits that are produced and whether seeds that are present in that fruits are viable or not. And we did kind of the opposite thing where we put a cage above the plants and we released the mosquitoes in that cage, and then took the mosquitoes out and identified them and saw whether they were able to remove pollinia from the flower and get some on their head. And what we found is that the fruit to flower ratio was higher when the plant was unbagged in the field. And when we had mosquitoes inside the cage, what we found is that it was like the fruit to flower ratio was higher when there were more than 2 plants in there, meaning that cross-pollination, so a mosquito moving pollen from one plant to the other, is better for the plant fitness. So now we know the mosquitoes get to that plant, we know that they're pollinated, but how do they locate that plant? And so the next step consisted in doing some scent collection and gas chromatography coupled with mass spectrometry analysis. And again, in the field, we bag the plants with the Teflon bags, and then we send some clean air through a charcoal filter, and all the volatile organic compounds that are emitted by the flowers are collected here in this trap. And then you can just elute that with hexane and you get your scent sample that then you can bring to the lab and analyze by GCMS. So on the right here, you have an example, a chromatogram for each of the 6 different species of orchid that we studied. If you're not familiar with chromatograms, the higher the peaks are, the more abundant the specific chemical is. And here you have the time. So just by looking very briefly through that, I'm not going to kind of give all of the list of the chemicals that are present in the scent, but you can see that they look kind of different. They emit like a very different amount of scent. So for example, the mosquito pollinated orchid has like kind of a very low emission rate compared to like these very fragrant ones such as Dilatata and Neuronensis. And if you take a look at some of these chemicals here that I highlighted by numbers, you can see that some of them are present in several species of orchids. That's the case for these like 12 to 14 that are present in Obtusata, Stricta, Dilatata, Neuronensis. And those are lilac aldehydes and lilac alcohols. And I'm going to talk a little bit about that later. But their abundance, again, is highly different. And so the question was basically, if they have some of these chemicals in common, like why the mosquitoes visiting this specific like P. obtusata plant and not the Stricta or Dilatata, for example. So if we take a look at like kind of a more like statistical representation of the scent, what you can see here, each dot is a single scent from the different species that we analyze. So they are color coded here. And what you can see is that they are clustering pretty well. But what is kind of striking is that the P. obtusata scent is like kind of pretty like kind of isolated from the very fragrant ones that are kind of clustering all together and which have like way more overlap in terms of their scent. And I want to highlight that this analysis is based on both the chemical compounds, so the different compounds that they have in their scent, but also their abundance. So the mosquitoes are smelling with their nose, their antennae, like we smell with our nose. And what we developed was this technique that is called Electron Tenograms for mosquitoes, where we analyze the electrical activity of the mosquito antennae in response to the different chemicals that are emitted by the flowers. And so these like couple of gas chromatography can give you like a clear idea of like what compounds in the scent, a complex like blend of chemicals present in a floral bouquet the mosquito can detect. So the mosquito's head is mounted between two electrodes, and the GC is going to send the chemical one by one to the prep. And each time you see this kind of deflection in the signal means that the mosquito can detect this specific chemical. So we did that technique with many different species of mosquitoes, as well as with various species of orchids, including ones that they are not visiting and not pollinating, such as Stricta and Neuronensis. And again, I'm not going to dive into the tiny details here. What I want you to see is that each time you see a black line going down, it means that the mosquitoes are actually able to detect chemicals. So that means that, yeah, all of these species are actually detecting many of the chemicals that are present in this scent, including from the plants that they are not pollinating and even not interested in visiting. Again, I look at statistical analysis here with the PCA, and what we found is that the responses of different species of the different species of mosquitoes for the Obtusata scent was kind of clustering like differently from what we got for the Stricta and the Neuronensis orchids. So based on the knowledge from the GCMS analysis, we created that artificial mixture that we then wanted to test as like a way maybe to use in the field for like a bait or in like a trap, for example. So we developed that blend, and we tested it using behavioral experiments. So that was like an olfactometer experiment where a mosquito is released here and asked to fly upwind and choose between one arm that is going to carry the odor of interest that you want to test, and the other arm is carrying a control, just like mineral oil, for example. And what we found for what we call snow mosquitoes, that's the group of mosquitoes that we get in the field. They're called snow mosquitoes because they are emerging when snow pools are melting. And what we found with these specific mosquitoes is that when you presented them with just like two controls, so that's what you can see here, they were like basically there was no preference between one of the two arms in the olfactometer, which is good because it means that there is no bias in our assay. I would maybe add that these results are presented as preference index, where a positive value means that they are attracted and a negative value means that they are repelled. So when we tested then the artificial mixture that we made, but without including the lilacs, there was a slight attraction. But when you actually added the lilac aldehydes to the mixture, then this attraction was higher, and it was the behavior on this response was as high as when you are actually testing the real plant. So means that our artificial blend is actually pretty good at like attracting these species. So I showed you that the mosquitoes smell with their antennae. So that's kind of the equivalent of our like nose, right? But how they process that scent and how they process like complex mixture in the environment is occurring at the level of the brain. And so here we got interested in these structures that are called antenna lobe. This is where they process the olfactory information. And that can be kind of related to our olfactory bulb. And by kind of analyzing the activity in these structures, then you can know basically like what exact compounds they are attracted to, and what is like in the blend like compounds that are like really exciting for these mosquitoes. So to do that, we did some two-photon microscopy. So here we developed that kind of holder where the mosquito is glued by the head, and then we perform like a tiny craniotomy to expose these two structures that are here, that are kind of grape shaped like. And each of these like kind of small structure, which is called a glomerulus, that you can see here, is getting input from olfactory receptors neuron that are found on the antennae. And so they all cluster in one very specific glomerulus. And then they project and send the information to higher processing centers that are then gonna drive the behavior consequently. So what did we see? So by monitoring the fluorescent activity in these different kind of structures, we were able to see what chemicals were really important and in mediating, to mediate like the attraction for the mosquitoes. So for this part of the work, we focused on these increpetus mosquitoes, and we got particularly interested and focused on two of these glomeruli, AM2 and LC2, as well as AL3, like in parallel. And so we looked at the fluorescence of these different structures in response to the P-obtusata scent that is rich in nonanal. And what we see is that it's mainly activating like the front part of the antenna lobe, that is AL3 and LC2. When you apply like aldehyde, it's kind of a mix between like all of these like glomeruli. But when you apply nonanal only, then all of a sudden it's like it's going to be mainly in the front part again. So there is kind of a tuning for like these different structures, and they are just like tuned to respond to very specific compounds. And here is just like a representation, a PCR representation of the different chemicals that we tested, and we wanted to see like how they were kind of orienting by like the responses that we got for the orchid mixtures and the orchid itself, itself. What I did not mention here is that this device that we developed allows us to do some recording at the level of the brain, but at the same time, you can also record the flight activity of the mosquitoes because they can fly on site. And we got also like flight responses of the different compounds that we tested along with the blend that we created. And the flight responses that we got for the orchid blend was as good as almost what we got when we were applying like a human odors. So this orchid mix is evoking like a very strong flight pattern as well as multiglomerular pattern in the brain. And so when we dived a little bit deeper, especially correlating that with what we found in the scent of the different orchid species, what we found is that the mosquito pollinated orchid, as the scent is dominated by nonanal, where when the three other orchids have mainly their scent dominated by lilacs. And so when we took a look at, again, our data for the two-photon analysis, what we saw is that this LC2 was really like tuned to respond to the P-obtusata scent and to nonanal, while here the AM2 was responding pretty well to the P-obtusata, the P stricta scent, as well as was really tuned to respond to the lilac. And so when we dive deeper and look at the responses of these different structures, these different glomeruli to the scent, what we can see is that really there is a fine tuning of LC2 to respond to nonanal and a fine tuning of AM2 to respond to the lilac. And so this ratio between the amount of lilac and the amount of non-anal that is going to matter, and it's going to be like really critical to mediate the attraction by the mosquitoes for these orchids. And this is what is also presented here. Again, this is behavior. So positive value means that they are attracted, negative means that they are repelled. In the case of the Pioptusata flowers, they were clearly attracted to that. In the case of the P. stricta, they were repelled, or for Uronensis and Dilatata, they were kind of indifferent. So this, like, again, this ratio is critical in mediating this attraction. So to summarize that part, we found that there are some differences in terms of composition and abundance for the scent of these different orchids. The mosquito is able to detect like a wide variety of chemical compounds present in the scent. But it's like the ratio of these compounds that is like clearly critical, and that explain why the mosquitoes are interested in visiting the Pioptusata and not the stricta, the Uronensis, or the Dilatata. So we did all of that work with mosquitoes that were coming from the field, but we were wondering, like, is that like kind of a conserve mechanism across like different mosquito species, for example? And we tested our blend with Aedes aegypti. That is a mosquito that doesn't overlap at all in terms of like geography with the orchids. So Pioptusata is in North America, and Aegypti is present in the US, and it's an invasive species, but it's really in the south of the US, and moreover, it's a species that is coming from Africa. So technically, they never evolved together. But when we tested whether they were able to feed on those plants or even be attracted, what we found is that definitely they are actually feeding on that plant, both males and females, and they are even able to get pollinia stuck on their heads. So even if these mosquitoes never like evolved with the plants, they are still able to get nectar from it and to pollinate it, which was kind of fascinating to us. So what about the chemical compounds that they can detect? So Aedes aegypti is detecting like a wide range, or almost all of the chemicals that are present in that scent. We also did this experiment with Anopheles stephensii, that is another major disease vector present in Asia and now invasive in Africa. They detect less, but they are still able to detect many chemicals in that scent. And when we did the two-photon analysis, we found the exact same results, where this like tuning for this specific like glomerulus or glomeruli is exactly the same, and they respond the exact same way as the snow mosquitoes. So this pattern of activation is a conserved thing across mosquito species. And if you look at the behavior here, again, the orchid scent evoked like a strong flight activity as well. Okay, so could we use that blend to kind of control Aedes aegypti mosquito population, because they are like problematic in many areas in the world because of the different like pathogens that they can transmit. So we did the olfactometer analysis again with Aedes aegypti, and here are like just like for you to remember the data that we got with the snow mosquitoes, and now looking at Aedes aegypti and Anopheles stephensii. The control did not evoke like anything. They were like, again, neutral in their responses. The Aedes aegypti didn't like really care about the lilac by itself, but it got repellent in the case of Anopheles stephensii. And when we presented the blend without the lilac, it was attractive for Anopheles stephensii and kind of neutral for Aedes aegypti. But in both species, the addition of lilac to the artificial mixture like really like attracted them. Like actually Anopheles stephensii was like kind of crazy about it. So there is kind of a chance here that we can use that scent to develop like new baits for mosquito control, and the next step would be to test it in the field directly, and we have a patent pending on that scent here. So the question remains, like, how can a mosquito originating from Africa be attracted to a plant that is going to be growing in North America? And how, by extension, like plants that we have in our backyards can influence mosquito fitness, in particular invasive mosquitoes in population dynamics. can we use that knowledge to inform our citizens and community? And can we leverage that knowledge to develop new tools for mosquito control by, for example, like creating baits and repellents? So let's move on to the second part of my talk, that is about ornamental plants and invasive mosquito species. So it all started by just taking a trip to the nursery and seeing all of those plants and thinking, whoa, could that be just like a big mosquito buffet? And so Ford Upshur, a PhD student in my lab, former MS student, now PhD student in my lab, is leading this project where he tested various plants that are commonly found in nurseries. As you can see, they are of different color, different shape. They emit different kind of scent, and they are also from different origin. And what Ford was interested in was to learn more about how invasive species, in particular Aedes aegypti and Aedes albopictus, adapt to local flora, and can we know a little bit more about the chemicals that are mediating these attractions, and can we use that knowledge for mosquito control? So the first thing that Ford did was to run plant vegetation assays where he had these plants in cages and basically released mosquitoes in there and recorded their activity with a camera. And taking a look at those videos, he was interested in learning more about the landing activity of these mosquitoes as well as feeding behavior. So for the landing, you have here in the green the Aedes aegypti mosquitoes, and the yellow are Aedes albopictus mosquitoes. And the landing for the different on the different kind of plants is illustrated here. And what you can see is that there is like a clear mosquito preference, but also a sex preference that is indicated by numbers that you find above the bars. So males and females are attracted differently to these plants. And some of them just don't attract many mosquitoes. That's the case, for example, of the red impatiens or the guar. But on the opposite, you have some plants that are attracting way more mosquitoes, such as the wave petunia, the goldenrod, or the yarrow. If we look now at feeding behavior, so this is again with like looking at videos, mosquitoes landed on many of these flowers, but actually didn't, like, never displayed any feeding behavior or any intention to feed, or like even like trying to feed, while others like really like the plants. So that's the case of the goldenrod that you can see here, and again the yarrow and the butterfly bush. So some plants were definitely more attractive than others. But do they get any carbohydrates from trying to feed on those plants? So to know more a little bit about that, Ford did this assay developed in the 80s, where you just crush the mosquito with this chemical and it gives you like this nice green color if the mosquito has like carbs in its gut. And for here you have the positive mosquito that were like positive sugar-fed mosquitoes for the different type of plants. And again, males and females are going to be illustrated in a second. What you can see is that, again, for some of these plants, such as like the marigold or the Mexican heather, like none of the mosquitoes that were released in those cages, we could not find traces of carbohydrates in their gut. So they couldn't get anything from these flowers. And on the opposite, you have some flowers that provided quite a lot of carbohydrates to these mosquitoes. That's the case of the goldenrod here or on the celosia. But if we stay on the case of the celosia, if we look at the proportion between males and females that were that had carbs in their gut, you can see that here in both species, Aedes aegypti and Aedes albopictus, only males like tested positive for this sugar. So there is like a clear also like difference between the way these plants attract males and females. So what kind of chemicals do they emit? So we run GCMS analysis with these different plants. And here's just like an example chromatogram for all of these plants. Again, I'm not going to dive in like detail for all of them. But what I want to show you is that they are all at the same scale. So some of them emitted quite a large amount of different type of chemicals. So that's the case of the goldenrod. You can see like many peaks. And that's also the case of the lantana. And some of others like did not emit that much. That's the case of this guar here that you can see. On top of that, the height of those peaks is very small, so that means that the concentration of those chemicals is actually pretty low if you compare again with the goldenrod or the lantana. So they are all pretty different, but they share some chemicals in common. You can see that I highlighted in red some of these plants that are here, and those are commonly sold as repellents in nurseries. Well, based on what we found here, some of those plants are definitely not repellent for the mosquitoes. They can land on it, and they can get carbs from them. So that's something that is important also to highlight in terms of how we can inform our citizens about good and bad plants to put in their backyard. So let's just dive deeper into two plants and analyze the scent of these two plants. So I took the goldenrod because that was kind of the one that was the most attractive for both species. And when we analyzed the scent, what was interesting is that first they made a lot of different chemicals, but there is no like one chemical that is going to be like kind of dominating the scent. It's really just a balance between many of those chemicals. And those, if you take a look at what they make the scent of, so this is like about 22% of beta-myrcene, the same amount almost for D-limonene, and again the same amount for germicrin D. But if you take a look at the literature, and that includes some work that we've done in the lab, all of those chemicals tested individually are like mostly repellent for mosquitoes. So again, it's like the importance of the ratio of these chemicals and basically the blend and the recipe, the cocktail that is making this scent that is going to be driving and mediating the mosquito attraction. So on the opposite here is a plant that the mosquitoes didn't really like. So that's something that you can put in your backyard if you want to, so you won't attract mosquitoes. And that plant, interestingly, had this like insanely big peak of one chemical. And this chemical is beta-ocimine. It dominated the scent by like almost 85%. And you can see that also this plant doesn't emit many other chemicals. And beta-ocimine is found in many different types of plants, including two others that we studied, marigold and lantana, that were both repellent for mosquitoes. But we know in the literature that they are really good at attracting bees and butterflies. So definitely like a bee-friendly or like pollinator-friendly plant to put in your backyard, not good for mosquitoes. But beta-ocimine, there is absolutely no work available on mosquitoes. And so that would be something that we are interested in learning more about, because if we can implement that chemical in a repellent, for example, that would be interesting. So we run an NMDS, again, analysis on these different scents that is based, again, on chemical compound and their relative abundance in the scent. And what was kind of interesting is that we got like we got a large blob of plants here that are kind of attracting a little bit the mosquitoes, but not that much. But the plants that were clearly repellent are clustered here. And the plant that was clearly like the most attractive one is here, the goldenrod. So this is kind of interesting to see that there is like really a trend of like what type of chemicals is making the scent and what behavior that you get when you do the analysis and what we can expect to see in the field. So in summary, ornamental plants can attract and feed two major disease vectors, Aedes aegypti and Aedes albopictus. And I want to highlight too that the flower morphology and the nectar access is important. And so that's the reason why in some plants we saw some landing activity, but they couldn't get any nectar from it because, for example, the flowers was too deep, and so the nectar is available only for butterflies that have like these long proboscis. As some species are attractive, some species are repellent, and definitely repellent labeled plants may not be that repellent. So what can we do now? Well, we can inform our citizens or like authorities on plants that can attract mosquitoes and should be avoided to plants in cities, for example, and that could clearly drive and help mosquito fitness and thriving. But we could also potentially, like if we analyze the scent of these different flowers, we could maybe predict whether, just based on the scent and our like analysis that I just showed you, using machine learning, see if we can predict whether those plants are going to be attractive or not based on just their scent. And obviously, there is like an impact of like using that knowledge for developing a bait that is attracting mosquitoes. And this is what Ford is currently doing with the goldenrod, trying to learn more about that plant and doing fieldwork this year. He found that it's attracting many mosquito species in the area. So talking about the area, what's going on in Virginia for sugar feeding? And we have also like several invasive mosquito species that we can focus on. So this work is led by Morgan van der Giesen, who is a master's student, and she's interested in learning more about how habitats and environmental factors affect sugar feeding in mosquitoes. And I should add that this is a collaboration between Luis Escobar in Fish and Wildlife and Clément Vinogier in biochemistry. And for this specific project, we're interested in learning more about the prevalence of sugar feeding in both urban settings and forested areas, as well as to know whether like a toxic sugar bait that I'm going to talk about in a couple of minutes can be effective. Obviously, the large question is how climate change will impact mosquito population in Virginia. That would be for another talk. So for this specific project, we collected mosquitoes every week from basically mid-March to the end of October at 10 different sites. So 5 were downtown Blacksburg, so urban sites. Forested site were Panda Pass and Mountain Lake Biological Station. So we are also like kind of 2 different kind of elevations. And we use these traps that are called BG Sentinel that attract host-seeking mosquitoes and host-seeking females primarily. So we identify all of the the mosquitoes that we collected, and we also measure their wing length for like more like in-depth like ecological like modeling later. So what this work can do is to, using predictive models to see how mosquitoes are going to be distributed in the future, especially in the context of climate change, and whether toxic sugar bait would be viable for the area. So I'm going to show you like only some of the results that we obtain for the sake of time, but here you have like a subset of the different species that we found at the different sites, so Mountain Lake, Panda Pass, and the urban area. Between again like kind of a phenology of the different species. And just by looking at that, what you can see it's some species are present in all of the different kind of habitats, and that's the case of this like invasive species Japonicus that you can see is both at Mountain Lake, Panda Pass, and in urban area, but in like a very low proportion. And on the opposite, you have this other invasive species, Albopictus, that is clearly mainly in urban settings. It's a very highly anthropophilic species, so it's highly adapted to biting humans, and that's kind of their preferred host. That gave us like a good indication of like the different type of flowers they could have access to. Obviously, in urban settings, you're gonna have access to more ornamental plants versus Mountain Lake, Biological Station, or Panda Pass, you're gonna have access to wildflowers mainly. We took also climatic data at each of these 10 sites, and we were particularly interested in temperature, like minimum, average, and max temperature, as well as humidity. And what we found is that there is a slightly higher temperature downtown versus the 2 other sites. It's slightly also drier, so that could also like kind of drive some of the mosquito assemblage that we find in these different habitats. If we look at the prevalence of sugar feeding in these different habitats, it's kind of highly variable, but what we can see is there are like trends that are the same in the 3 different habitats. So it's starting about the same at lower elevation. So the rate here, the percentage of mosquitoes that are sugar fed is higher at the beginning of the season. The reason why is that it's when the first flowers are blooming, spring, and also these mosquitoes are coming out either of diapause or they are just emerging. So they are really, really excited by getting some of the sugars that are available in those flowers. And then there is this second peak that is more at the beginning of the summer that is linked to the presence of fruits and more flowers and more trees blooming in particular. But what you can see is that the like total percentage or like average percentage of sugar feeding in these different habitats is quite different. At Mountain Lake, it was quite high, and one reason could be due to the fact that we collected fewer mosquitoes there, and because the mosquitoes that are present at that site don't really like the trap that we deployed. On the opposite, at Panda Pass, we collected way more mosquitoes from one of the species that is probably consuming less sugar. And then the urban setting, we collected many of these Albopictus mosquitoes that seem to be kind of quite attracted by the sugars that are provided by ornamental plants. So to summarize that part, we know that sugar feeding is going to be both happening in urban and forested habitats, and that these species, whether they are local or invasive, can feed on those plants. And so it's worth it, like trying to develop like a toxic sugar bait and to try to test it. That there is like a clear also seasonality for the sugar feeding in these different areas. So how can we use that to control mosquito populations in Virginia? So that brings me to the fourth and last part of my talk, developing a toxic sugar bait for Ochleotatus japonicus. So what is that mosquito? You might not have heard about it. That's an Aedes species, and it's another of the invasive species that we have here in the US. And it's highly invasive pretty much everywhere in the world, and in Europe, in New Zealand, and like different countries around the world. Basically everywhere it gets, it can just like spread. And the reason why is that it has like this important ecological plasticity. They are very cold tolerant, they are very large mosquitoes, and they have also this kind of diverse range of preferred hosts. So they can feed on mammals, they can feed on birds. The bad thing is that they have been shown to be competent for transmitting dengue and chikungunya viruses. And this project is led by Lauren Frislevich and Ash Van Winkle in my lab. And their objective was to develop a bait and later a trap to reduce these mosquito populations. So what is a toxic sugar bait? So ATSB stands for attractive. So A as attractive, you put an attractive odor in there. So that can be like a fruity odor or like a plant odor. You're gonna put a toxic chemical in there. So in that case, what Ash and Lauren tested was boric acid. You need a phagostimulant, so you use sugar because this is what they get from flowers, and you can bait both males and females. Again, that's kind of really interesting in terms of pest management. You can also use a dye, so you can monitor whether mosquitoes are feeding on it, and that can come in like diverse forms, from like these bottles that can be like placed in indoor or outdoors. You can also have that on like cloths that are suspended, and some people are also spreading vegetation with these toxic sugar baits, and it seems to be working very well. And the good thing is that there are so many like good like things about these traps or like these baits, because they are first very inexpensive to develop, they are sustainable, they are environmental friendly, they are very species specific if you use the right trap and the right compound that is emitted by that trap or that bait. And it is efficient like against a wide range of species. But if it has been shown to work with like Anopheles species and Aedes species, different kind, nothing is known about Ochleotatus japonicus. And so this is why Lauren and Ash focused on that species. And so they conducted this cup assay where they release mosquito in those cups and they provide a cotton balls imbibe with the solution that contains or not the toxic chemical. And so they monitor the survival of both males and females. And this is what you can see here. So you have the survival in both females and males through time. So the assay lasts 4 days. This is what we can expect the product to stay alive and kind of good in the field. And what you can see here is the plain bars are the control. So there is no toxin in there, no toxic chemical. And what you can see here in the bar that are like pattern, those bars are representing data that is contain the boric acid. And what you can see is that after like about 2 days, most of the mosquitoes, whether they are males or females, are dead if they're imbibing this boric acid solution. So the boric acid is working. So let's move on and adding like an attractant to the solution. So the first fruit that Lauren and Ash tested was mango. It has been used in many different species, in particular in tropical species. And they got the exact same results. They like it, and they die if there is boric acid in it. same results are almost obtained with peach. So what about fruits that are gonna be available for the mosquitoes towards the end of the season, like before basically they die or like they enter diapause, for example? So using blueberry, they also like this one very well and eat that very well and died like even faster in less than 48 hours. The same thing for the blackberry. I think this part here is the one that we are still conducting. Like we need to increase the numbers, but they seem to be like liking this solution as well. And then the last one that we tested was kind of starting from the idea that in some countries it's really difficult to find those fruits. It's difficult to find these different compounds that are found in the toxic sugar beet. So one crazy idea was to use soda. And the reason why is because also we know that bees, for example, are really attracted to these sodas and can feed on them. And it seems that it's working very well for the mosquitoes too. Here in the case of males, like they didn't even last 24 hours. They just basically died of food coma by soda ingestion. But the soda itself is not killing the mosquitoes, which is good. You can see that after 4 days, they're all alive, which is probably a good news for us that actually drink that. So to summarize that part, toxic sugar beet is effective against Ochlorotatus japonicus for both males and females. And here, as you can see, we have multiple options available to deploy that in the field. And that's gonna be kind of the next step for us to conducting some field tests and making sure that it's gonna be species specific. So, and avoiding like off-target species basically. And this is all in the trap design. So I hope that I've convinced you that it's not only important to study blood feeding behavior and host pathogens and mosquito host interactions, but also focus on sugar feeding that has been like, definitely historically overlooked. So yeah, and I would like to thank all people that were involved in that work, current collaborator here at VT as well as outside in the US and in the world. I want to thank particularly people who are involved in Morgan's project, so Clément Vinogi in biochemistry, as well as Luis Escobar and Mariana Castadena Guzman, who are working with us on this Global Change Center funded project. I also want to thank other people for their support as well as funding sources. And a big shout out to my lab members. Thank you all for everything that you do. It's a great pleasure to work with all of you. And I also want to say happy birthday, lab, because we exactly started 3 years ago. So yeah, happy birthday, everybody. And past and present, obviously, like many undergrads also participated in previous projects. And last but not least, I would like to read this land acknowledgement. So we acknowledge the Tutelo Monacan people who are the traditional custodians of the land on which we work and live, and recognize their continuing connection to the land, water, and air that Virginia Tech consumes. We pay respect to the Tutelo Monacan nations and their elders, past, present, and emerging. Thank you very much, and I would be happy to take any question.
Thank you very much, Chloe. It was really super cool talk. We have a couple of questions here. Let's start with the one from Felicia Edgecombe. She has a question regarding the chemical mechanisms for which these insects recognize sense that they're repellent or not. So she says, do you have any sense of the chemical mechanism for specific compounds being repellent? Are they toxic to the mosquito? What is it now?
Well, right now, I don't think that we do. We know that some of these chemicals cannot be detected by the mosquitoes just because they basically don't have the machinery. But the way that works exactly, I think, is just getting like unraveled. And this is not something that we are really working on, but that's a very good question.
I have a question regarding the essence itself. Is this, do the plants have this essence all year long or seasonal, or is, is always present? Does it change with humidity, temperature? How does it work?
Yeah, definitely. You're right. It's gonna change during the day. Some plants are emitting more scent during the night, for example. So that's the case of petunia, because their pollinators are nocturnal. So they are producing more scent at night to basically just drag them to them to be pollinated. So there is like daily variation in the scent emission of many plants, and there is also like factors that are affecting the scent production. So temperature, humidity, if it's drier, it's going to be more concentrated, for example. And definitely towards like when they are at the end of the blooming, so when they don't need like those pollinators, you know, anymore, they reduce the scent, or the scent is going to change and even like become like a repellent. That's what we shown. I didn't see that today, but with the orchid, when you go like towards the end of the season, they produce like more of like specific compounds that are just like repelling the mosquitoes because they just don't need them anymore.
Yeah, exactly. We have one more question from Felicia. It says, what concentrations of boric acid and sugar do the traps use?
So for the sugar, I think that's like pretty high. It's going to be like about 20%. And I think 1% boric acid is enough to kill all of them. So it's not that much.
Okay. Sorry, I have another question. I couldn't avoid looking at your preference index. And based on your description, I assume that the preference index will be 0 or 1, right? I mean, it's just you don't go or you go or you don't go in that direction. But I couldn't avoid noticing that your preference index has numbers like 0.3 or 0.5. So how did that come?
So it's basically considering also mosquitoes that were not attracted to that scent. So it's just to illustrate that, yeah, exactly.
Yeah, that they stay there. Okay.
Yeah.
Okay, that makes sense.
So it's based on mosquitoes that are attracted versus the total number of mosquitoes divided by the total number of mosquitoes. So it's kind of like a weird calculation that we make, but it's, you need to kind of highlight the mosquitoes that didn't want to come to your like scent basically.
Okay, we have a question from Aaron. Nice talk, Chloe. I'm just pretending to be him. Do you know if there is a crosstalk between AM2 and LC2 in the brain? In addition to calcium, are there any changes in the electrical activity?
Can you repeat the beginning?
Yes. Do you know if there is a crosstalk between AM2 and LC2 in the brain? Let's go with that one first.
We don't know. That's a very good question. No idea. I think we would need to dive deeper in that. And I think one of the collaborators on that project is interested in answering that kind of question. So hopefully we'll have that response soon. But we are like, everything that is with like mosquito olfaction and like the structure of the antenna lobe and how that like circuitry is just like just happening right now. So yeah, many questions are still like kind of to answer.
He has a follow-up question, if you don't mind. In addition to calcium, are there any changes in the electrical activity?
I don't think so. I mean, not that we have like recorded. So yeah, we just based our like all of our measurements were mainly like fluorescence based on like calcium variations. But so
I'm just gonna ask another questions until someone else pop up. Anopheles, right? I mean, it seems like an obvious thing that I'm pretty sure someone already tried is to use some of this essence as repellent for Anopheles, right? It will be a very effective mechanism, cost-effective mechanism for reducing malaria, for example. Well, what's going on? Why is that we don't have one of those, or we do have one and I ignore?
Yeah, we do have some compounds that are working very well in the field that have been tested against like Anopheles gambiae, for example, in Africa. So some people are just like basically mixing mango with sugar in those bottles to attract the mosquitoes outside of their home. So yeah, I mean, some of them are existing for the like malaria mosquitoes, definitely, and they're working very well.
Oh, wow. Okay.
And those are just like really not expensive to
exactly.
produce. So that's kind of
it's a very cost-effective mechanism to actually contribute to the reduction of the mosquito population in those.
Yeah, exactly. The cheaper the better here, for sure.
Exactly. Is there any other question for Chloé? If not, thank you very much. I'm very happy to finally hear your presentation.
Thank you so much, Carla, for inviting me. It was a great pleasure, and thank you, everybody.
Okay, no problem. See you guys next Friday. Take care.
Bye.